Welcome to the next chapter. It is the cell. Now, animals and plants' bodies are made up of small microscopic units called the cells. The cells make up the structures of the living organism and are responsible for carrying out the various biological processes. Some organisms are composed of a single cell only and are said to be unicellular, for example, the amoeba and bacteria. Others are composed of many cells and are said to be multicellular, for example, humans and giant trees. Therefore, a cell is defined as the basic unit of life or the basic unit of an organism. The cell is a very small structure and cannot be seen with the naked eye, and hence it requires the use of a powerful magnifying instrument called the microscope. In 1650, Zacharias Janssen invented the compound microscope, which combines two lenses for greater magnification. Later on, in 1665, Robert Hooke used an improved compound microscope to observe the cells. Later, Antony van Leeuwenhoek developed a better microscope with lenses which provided greater magnification. He used this instrument to observe the nuclei and unicellular organisms, including bacteria. In the 1930s, the electron microscope was developed, enabling much more of the internal structures of the cells to be observed. Now, since then to date, the electron microscope has been widely used in biology, showing more details in the internal structures of the cells. The light microscope. Now, in studying biology, some small objects require to be magnified so that details can be revealed. A hand lens could be used, but in some cases, there are finer details that require a microscope to be used. A microscope is designed to give finer details of an object that would otherwise not be seen by the naked eye or a hand lens. The commonly used microscope in schools is the light microscope. It is called a light microscope because it uses light for illumination of the specimen to be viewed. A microscope is a delicate and expensive instrument and should therefore be handled with care. It is therefore important to know the parts of a microscope and how to use and care for it. Handling of a microscope. When using a microscope, the following should be observed. Always use two hands when carrying a microscope. One hand should hold the base to provide support, while the other hand holds the limb. One should never place the microscope too close to the edge of the working bench or the table. Do not touch the mirror and the lenses with your fingers. Dirty lenses should be cleaned using a special soft lens tissue paper or tissue paper moistened with ethanol. Other parts of the microscope may be cleaned using a soft cloth or tissue paper. Do not wet any part of the microscope and make sure the low power objective lens clicks into position in line with the eyepiece before and after use. After use, always clean and store the microscope in a safe place free from moisture and dust. Parts of the microscope. Now, the light microscope is shown on your screen and the various parts are shown here. We have the eyepiece, the coarse adjustment knob, the body tube, fine adjustment knobs, the arm or the limb, the hinge screw. We also have the objective lens, revolving nosepiece, clip, stage, the condenser, the diaphragm, and the mirror, also the base. Now, what are the uses and functions of these parts of the microscope? The eyepiece. The eyepiece is the part closest to your eyes through which you look. It contains the ocular lens, which magnifies the image produced by the objective lens. It is often labeled with its magnification, for example 5x or 10x, indicating the number of times it magnifies. The body tube holds the eyepiece and the revolving nosepiece. The arm or the limb is the curved metal piece. It holds the body tube in place over the stage and the base. The revolving nosepiece holds the objective lenses in place, enabling change from one objective lens to the other. The objective lens brings the object into fine focus and magnifies it. Each objective has a lens and a tube-like holder. The longest tube holds the high power lens. The smallest holds the low power lenses. Some microscopes also have a medium power lens. The stage is a flat platform where a specimen on the slide is placed. It has two clips to hold the slide in position. The condenser concentrates light on the object on the stage. The diaphragm is an aperture that regulates the amount of light passing through the condenser to illuminate the specimen. The coarse adjustment knob raises and lowers the body tube for longer distances to bring the image into fine focus. The fine adjustment knob raises and lowers the body tube through smaller distances to bring the image into sharper focus. It is mostly used with a high power objective lens. The mirror reflects light through the condenser to the object. In certain cases, some microscopes would often have a light bulb in place of a mirror and would therefore need to be connected to a power source. The base provides firm and steady support. The substage adjustment knob raises and lowers the condenser. Now place the microscope on the bench with the stage facing away from you, so that the mirror faces the direction of light. But if the microscope has its own source of light, connect it to a power source and switch it on. Check that the condenser lens is just below the stage. Put the objective lens on the low power and move the stage to its highest position. Adjust the light to the medium or medium-high range. The highest light does not necessarily mean the best image. You can always adjust this light later on. If you are using a mirror, set the mirror so that enough light is reflected to the stage. The microscope is now ready for use. Open the clip with one hand and slide the microscope slide towards the back of the stage. Use the knobs under the stage to move the slide back and forth, right and left. Now, starting with the low power objective lens, look through the eyepiece. Focus on the image, which should be nearly focused. Once you focus the image, move it to the field of view. Then move to the medium power objective lens and refocus. Use the fine adjustment knob to find the fine focus. insert the specimen and move to the highest power objective lens and refocus. Magnification. Now, magnification of the object viewed is calculated by multiplying the eyepiece lens magnification by the objective lens magnification. If you look at the light microscope, you notice the magnification is written on the eyepiece and also on the objective lenses. So when you multiply the magnification on the eyepiece by that of the objective lens indicated, you get the total magnification. For example, if the eyepiece lens has a magnification of times 5 and the low power objective lens has a magnification of times 15, then the total magnification is 5 times 15, which gives us 75. This is the number of times the detailed structure being viewed has been magnified by the microscope. The electron microscope. Now, the electron microscope is more powerful and more sophisticated than the light microscope. It has more complicated parts. The electron microscope can magnify up to 500,000 times and has a higher resolving power. The high resolving power of the electron microscope enables it to make out as separate things, objects which lie close to one another. The high resolving power makes the electron microscope an important tool in research. Electron microscope uses a beam of electrons instead of light to illuminate the object. Research scientists with the aid of an electron microscope have prepared some of the detailed electron micrographs that will be observed in the study of the cell. Now, while using the light microscope, you may have been provided in the school laboratory with permanent slides of plants and animal cells, and the technician or the teacher requires you to observe them. When you use the light microscope, some of the cell structures can be seen, and these structures might include the cell wall, cell membrane, the cytoplasm, the nucleus, vacuole, chloroplast, and many others. Now, the structures within cells are called organelles, and they perform specific functions within a cell. So let's look at the functions of various organelles within a cell. The cell membrane. The cell membrane is also known as the plasma membrane or the plasma lemma. It is seen to consist of 3 layers under the electron microscope. The 3 layers are composed of 1 layer of phospholipids sandwiched between 2 protein layers. The cell membrane is flexible and has pores. The main functions of the cell membrane include: the 3 layers give it strength to enclose the cell content. The spores allow movement of particles in and out of the cell. The phospholipid layer controls the movement of water-soluble and fat-soluble molecules in and out of the cell. The cell membrane is thus said to be selectively permeable or semi-permeable. Cytoplasm. Cytoplasm consists of a fluid medium in which chemical reactions take place. It contains organelles and other inclusions such as starch, glycogen, fat droplets, and many other dissolved structures. Inclusions are non-living structures within a cell like glycogen. The cytoplasm is not static but has a movement known as cytoplasmic streaming. Mitochondria. In singular, it is called a mitochondrion. Mitochondria are sausage-shaped and are sites for respiratory reactions that yield energy for the cell. Mitochondria are bound by 2 membranes. The inner membrane is greatly folded into crusti to increase the surface area for respiration. The arrangement and number of mitochondria in a cell depends on the cell's energy requirements. Cells which require large amounts of energy contain many mitochondria. Such cells include the muscle cell, sperm cells, apical meristems, and the kidney cells. Endoplasmic reticulum. Endoplasmic reticulum appears as a series of interconnected channels running throughout the cytoplasm as shown on your screen. It is continuous with the outer membrane of the nuclear membrane. Some endoplasmic reticula has granules called ribosomes on their surfaces and are therefore referred to as rough endoplasmic reticula. The others are not associated with ribosomes and are thus referred to as smooth endoplasmic reticula. The rough endoplasmic reticulum transports proteins while the smooth endoplasmic reticulum transports lipids. Ribosomes. Ribosomes are spherical in shape. Some are scattered within the cytoplasm while others are bound to the surface of the rough endoplasmic reticulum. They are sites for protein synthesis. Lysosomes. Lysosomes are spherical sac-like organelles bound by a single membrane. They contain lytic enzymes which break down large molecules, destroy worn-out organelles, or even the entire cell. Golgi bodies or Golgi apparatus. They are found close to the cell membrane. Their functions are packaging and transport of glycoproteins. They are often involved in secretion of synthesized proteins and carbohydrates. Centrioles. Centrioles are rod-shaped structures located just outside the nuclear membrane. They take part in cell division and in the formation of cilia and flagella in cells where these structures occur. Plant cells have no centrioles. Chloroplasts. Chloroplasts occur only in plants. Chloroplasts are egg-shaped. They are surrounded by two membranes and contain a gel-like stroma through which runs a system of membranes that are stuck together to form a grana. The granum contains chlorophyll. The chlorophyll traps light energy that is used during photosynthesis. Chloroplast is the organelle in which photosynthesis takes place. Vacuoles. Vacuoles are sacs that are filled with fluid called cell sap. They vary in shape. Animal cells contain small vacuoles, while in plant cells the vacuoles are larger. Sap vacuoles store sugar and salts, therefore contribute to the osmotic properties of the cell. In some unicellular organisms, food vacuoles store digested food while contractile vacuoles excrete unwanted materials from the cell. Cell wall. The cell wall is the rigid outer cover of the plant cell and some lower organisms. In plants, it is composed of cellulose fibers. It gives the plant cell a definite shape as well as providing mechanical support and protection against mechanical injury. The cell wall allows gases, water, and other substances to pass through it. Nucleus. Under the electron microscope, the nucleus is seen to have double membrane called a nuclear membrane. The membrane has pores which allow the passage of materials in and out of the nucleus. The nucleus is made of viscous fluid called the nucleoplasm, in which the nucleolus and chromatin materials are suspended. The nucleus controls all the activities of the cell. The nucleolus is responsible for the manufacture of ribosomes, while chromatin contains hereditary material. Comparison between plant and animal cells. Plants and animal cells have a lot of similarities in their structures and functions. They, however, have several differences between them. The plant cells are usually larger compared to the animal cell. The plant cell also have a regular shape, while that of the animal is irregular. Plant cells have a cell wall in addition to the plasma membrane, while animal cells only have the plasma membrane. The plant cells usually have larger central vacuole, while animal cells usually have no vacuoles, but when present, they are temporary and small structures within the cytoplasm. The cytoplasm of the plant cell and the nucleus are usually located towards the periphery. The cytoplasm of the animal cell occupies most space in the cell, with the nucleus usually located in the center. Some plant cells have chloroplast, while animal cells absolutely have no chloroplast. Plant cells usually store starch, oils, and proteins. Animal cells store glycogen and fat. Plant cells have no centriole, while animal cells do. Cell specializations. Now, cells are structurally differentiated or modified to perform specific functions. Such cells are said to be specialized. For example, the root hair cell has an extended surface for absorption. A sperm cell has a tail-like extension for swimming towards the ovum. Now, a group of cells that are specialized to perform a particular function make up what is called a tissue. Different tissues that perform related functions form an organ, and organs form what is called an organ system. Tissues. These are cells of a particular type that are grouped together, and they perform the same function. Types of animal tissues. Epithelial tissue. Now, this is a thin continuous layer of cells for lining and protection of the internal and external surfaces. Blood tissue. Blood is a fluid containing red blood cells, white blood cells, and platelets. Blood transports many substances, including gases, nutrients, and protects the body against infection. Connective tissue. Connective tissue consists of strong fibers that connect other tissues and organs, holding them together. Types of tissues in plants. We have the epidermal tissue of a leaf, and this is a single thin layer of cells covering the outer surface. It protects the inner tissues of plants from mechanical damage and infection. Palisade tissue, and this is a group of cells rich in chloroplast containing chlorophyll. It has site for absorption of light, energy, and manufacture of food by photosynthesis. Conducting tissue, or the vascular bundle, and this consists of xylem and phloem. Xylem conducts water and the dissolved mineral salts in a plant. Phloem conducts food substances in solution. Organs. Now, various tissues occur in living organisms. These tissues may be specialized and grouped together to form a functional unit called an organ. In plants, the organs include roots, leaves, flowers, and stem. In animals, the main organs include the heart, brain, kidney, lungs, liver, and so on. Organ systems. Several organs whose functions are coordinated and synchronized to realize an effective action in a living organism is referred to as an organ system. In animals, some of the organ systems are digestive, circulatory, excretory, respiratory, and many others. For example, the digestive system is composed of the organs like the esophagus, stomach, intestines, and associated glands. The organ system is sometimes referred to as simply as system.
Without many of these discoveries and inventions, few of us would be alive today. Welcome to WatchMojo.com, and today we're counting down our picks for the top 10 greatest medical breakthroughs in history. Number 9. The birth control pill. Both the medical and social spheres of human life were significantly affected with the introduction of the birth control pill in the 1960s. By preventing unwanted pregnancies, the contraceptive displayed the great potential of medicine and gave women power over their own lives. The pill also helped ensure eventual salary parity between the sexes and indelibly aided the feminist revolution.